So in this lecture, we're going to be discussing white blood cells and platelets, and we'll start out with white blood cells. Now, white blood cells, they are the smallest fraction of, and the platelets too, they're kind of included in that Buffy coat. They're the smallest fraction of the blood. Excuse me, they only make up about 1% of total blood volume. And if you were to collect a microliter, that's 1/1,000,000th of a liter of blood, and then count the number of white blood cells in it, on average, somewhere between 4,500 and about 10,000 of them per microliter. And they're the only formed elements that have a nucleus. And you can see in these images here, the darkly stained portion of the cell, that is the nucleus. And the nuclei are kind of unusual in white blood cells. Their function is to protect the body from disease. And we're not really going to dive deeply into that function until we get to Chapter 16, the lymphatic system. But like red blood cells, they are stimulated by hormones to be produced. And there are two groups of hormones that do this, the interleukins and the colony-stimulating factors. And we're not going to say much more about that, but again, negative feedback is involved in the control of white blood cell productions. So we can classify leukocytes, the white blood cells, into two categories, and that's really based on the appearance after a stain has been applied to them, and we look at them under the microscope. And so the two types are the granulocytes, and these are ones that when they are stained, there's this real grainy appearance, and you can see it there. That grainy appearance there is because of the large organelles that are found in them that accept that stain. And they are primarily lysosomes, and if you recall from Chapter 3, lysosomes are the little packets of enzymes that are used to destroy things. And they also are secretory granules, so these little packets, membrane-enclosed packets of enzymes that are released from the cell. So the granulocytes have this real, very grainy appearance. The agranulocytes, and I should have said, this is also a granulocyte here. You can see lots of granules in there. The agranulocytes have a much smoother appearance when they're stained because the organelles, the lysosomes and these secretory granules, while they have them, they are much smaller, and so they are less obvious when looked at under the microscopes. So neutrophils are granulocytes. Neutrophils are the most abundant of white blood cells. In circulation, they make up a half to slightly more than half of all white blood cells that are moving around in the blood. When you look at the nucleus, it has this bizarre kind of multi-lobed appearance. So these are not multiple nuclei here. These are just lobes of the nuclei. And sometimes they are kind of bound together or attached to each other by these long filaments, like you see here. And there's no real specific shape to the nucleus, and so they are sometimes called polymorphonuclear leukocytes. How's that? I promise you, that will not be on the exam, so you don't have to remember that. But the idea is that poly means many, morpho means shape, so they are the many-shaped nucleuses, or the leukocytes with many-shaped nuclei. So neutrophils will take up two kinds of stain, acidic stain and basic stain. And so when you look at them stained with these two stains, the acidic and basic stains, because the acidic stain is red and the basic stain is blue, they have a light purple color under the microscope. So as part of examining somebody's blood and looking at the white blood cells, you would apply both of these stains together, well, separately, one at a time, onto the same slide. But when you're done, then and look at them under the microscope, because they accept both acidic and basic stains, they have this purple color. The function of the neutrophils is to phagocytize bacteria, fungi, and sometimes they will even phagocytize viruses. So these neutrophils, they are just like, while they're relatively small cells, they have a big function as phagocytizers, and they will be the first of the nuclei to arrive. I'm sorry, of the white blood cells to arrive at an infection, because there's so many and they're constantly on the prowl looking for bacteria or fungi that they can take over. Short-lived, because they're rapid consumers, but they only have enough stuff inside them to take out between 12 and 1 to 2 dozen bacteria. So once they have consumed their allotment, used up all of the enzymes that they've made to destroy the things that they consume, then they die, and they are the primary component of pus. So all that whitish goo that squeezes out of a sore is primarily, or has a lot of that material, is the white blood cells. Okay, so neutrophils, all about consuming bacteria and fungi and taking care of infections when they arrive. Eosinophils, they take up a stain called eosin. So it's a reddish colored stain. It's the acidic stain that is used. And so because they take up so much eosin, then they've been named eosinophils. That means they like eosin. Phil, if something is philic, remember, it means it's liking. So these are the eosin-liking stains. They take up that deep red coloration, and there we are. So when you see a red stained granulocyte, you know you've got an eosinophil. They have bilobed nuclei. That means 2 lobes to the nucleus. So here's one lobe, here's the other lobe in this eosinophil. Again, they're still connected to each other. They're not 2 nuclei, just 2 lobes to it. These guys are involved in moderating allergic reactions. And so their job is to go around and inactivate histamines that have been released. So if you recall back when we talked about inflammation, histamines were part of what causes inflammation. So when somebody has a really extreme allergic response, it's the primary cause of that is this great release of histamine that occurs. And so the eosinophils, they will go around gobbling up histamines, preventing the allergic reaction from going out of control. They also defend against parasites. So if a parasite is found in the body, they will cozy up to that parasite and then release the granules of digestive enzymes, and the enzymes will then take out the parasite, hopefully. Now, many parasites defend themselves against this, but that's the goal. They make up only a small part of circulating white blood cells, somewhere between 1 and 3% in a healthy person. Basophils, as their name suggests, will take up the basic stain, and so they take on a dark blue coloration when stained. They also have a bilobed nucleus, and these guys will move around from the blood into damaged tissues, and they will release histamines as well as, along with the mast cells. So they help to stimulate inflammation, and then they also release heparin, which is a chemical, an enzyme that inhibits clotting. So heparin will help to keep the blood from clotting. So the point of the inflammation that basophils help to stimulate is exactly the same point as the inflammation caused by mast cells as they release histamines and heparin, and they increase the blood flow to an area, and then kind of all that swelling, fluid release will cause swelling in the area, and that helps to kind of quarantine maybe because of all the pressure of the swelling, helps to quarantine the area and slow the spread of microorganisms, so that all of that area, there's opportunity then for, say, neutrophils to move in and take out all of the bacteria that are causing the infection. Okay, so that's neutrophils, eosinophils, and basophils. So what about the agranulocytes? Two kinds of agranulocytes. There are the monocytes. These are the largest of the white blood cells. They are also polymorphonuclear because the nucleus could be spherical, it might be oval, or it could be lobed. They comprise something between 3 and 9%, so again, a relatively low proportion of circulating white blood cells. But after they've circulated for about 24 hours, then these dudes will migrate out of the blood and into the tissues. And once they're found there in the tissues, they crawl around and act and are very phagocytic. So they are referred to. We change their name, and this is because it wasn't recognized that monocytes and macrophages were the same cells, just in different locations. So when we first found them in the tissues, we saw this large wandering phagocytic cell and called it a macrophage. So that name change that's why, is because we didn't recognize at first that these were two different kinds of, or that these two different cells were actually the same cell. So they're called monocytes when they're in the blood, macrophages when they're out there in the tissue, and their job is to phagocytize bacteria, any dead cells that they run across, and other debris. So if you're injured and cells are starting to die, or they've just dumped out all of their cell contents, then macrophages will come along and they will clean all that stuff up. Okay, so monocytes are one of the circulating agranulocytes, and the other type of agranulocytes that circulate in the blood are called lymphocytes, and we have multiple kinds of lymphocytes. These are cells with a very large and spherical nucleus and only a little bit of cytoplasm, so they're pretty easy to recognize and distinguish from the others because all of the other leukocytes have a lot more cytoplasm than lymphocytes have. And these dudes are the ones that provide our immune response. So these are the ones that we're going to be talking about primarily when we get into Chapter 16. Two types of lymphocytes. There are T cells. T cells will go around and directly attack microbes, tumor cells, and if let's say someone receives a transplant and we start to have that reaction against the transplanted materials, then it's T cells that are going up and directly trying to kill the transplanted cells. So T cells interact directly with cells and kill them off. B cells are the second kind of lymphocyte. B cells have a different job. Rather than directly attacking cells, they do this more indirectly. They produce antibodies against foreign materials, and so these antibodies then circulate in the blood, and the antibodies will bind to foreign materials and the ones that they are designed to go and attack. So these antibodies will bind to that foreign material, and then that serves as a signal to T cells and to other immune system cells to go out and kill them. So lymphocytes are the other high percentage white blood cell that are found out there. So if somewhere between 50 and 60% of the white blood cells are neutrophils, somewhere between 25 and 33% of the circulating white blood cells are lymphocytes. So everybody else is small potatoes percentage-wise in the circulating in the blood. Okay, so what is it the white blood cells are doing? They travel around. They're carried in circulating blood And they can also like reach out, glom onto the side of a blood vessel, and then crawl against the current. Let's see. So if they're trying to find a chemical signal that's being released by damaged tissues, for instance, they might go past, they slide past where that site of damage is. They get that big hit of chemicals being released by the damaged tissues. They can slow themselves by grabbing a hold of the side of the capillary wall and then crawl backward by amoeboid motion. So that's how amoeba crawl around. And I think I showed you last semester, if you were in my class, showed some video of amoeboid motion where these white blood cells are crawling around by streaming out a chunk of their cytoplasm into a portion of the cell membrane and creating little legs that crawl them forward. So, if this is a white blood cell, they can, let's see if I can somehow kind of capture it. Like, ooh, we're going to go in this direction, and pretty soon the whole, all of the white blood cell is moving in that direction. So. Okay, so that's what amoeboid motion might look like as it's crawling and the cytoplasm is streaming into this pseudopod. They can also do something called diapedesis, where, if you recall, we talked about that? No, we haven't. We've talked about capillaries being a little bit leaky, I think, in the past. So here you can see a zone where there's a little gap that allows fluids to exchange very easily between the blood and the extracellular fluids that's around that capillary. Well, white blood cells are capable of squeezing through those little gaps, and that function, that squeezing through and getting into the tissues or into tissue fluids, is called diapedesis. And one of the ways that I remember it is that ped means foot, and so they're going to go on walkabout, if you will. So they go and travel amongst the fluids of the peripheral tissues. And neutrophils are so they have the capacity to do movement. They can respond to chemicals. So neutrophils especially are attracted by chemicals that let them know that there's damage here and that we need to go and take out the bacteria or the parasites. I'm sorry, the bacteria or whatever, the fungus, that's what I was looking for, that are causing that damage. White blood cells are capable of phagocytosis, especially neutrophils and monocytes. We call them, again, macrophages when they're out there in the peripheral tissues crawling around and eating stuff. So phagocytosis is really, really important. And remember, neutrophils will fill up with the digested foreign material, then they die. Macrophages can come along and clean them up. And then last of all, basophils have the capacity to secrete histamines and heparins in damaged tissues. So movement, being attracted to chemicals, phagocytosis, and secretion are all functions of white blood cells. What do the counts look like? Well, we talked about how 4,500 to 10,000 per microliter is a common, average count. Anything above 10,000 then is referred to as leukocytosis. Anytime you see cytosis, that means excessive cell counts for something. So leukocytosis is greater than 10,000. Leukopenia, anytime you see penia, that's going to mean deficiency in. So less than 4,500 is referred to as leukopenia. So leukocytosis often indicates some kind of acute infection. So a bacterial infection, a fungal infection, or even a viral infection. Leukopenia, on the other hand, may accompany fever. Viral diseases frequently result in leukopenia, or they can indicate anemia or heavy metal poisoning. So leukocytosis, excessive white blood cell counts. Leukopenia, excessive deficiency in white blood cells. Leukemia is a cancer of the hematopoietic stem cells that are producing leukocytes, and you'll see really, really extreme counts, 100,000 per microliter. So, an order of magnitude more than what might be normal. And then we can use the count of white blood cells to help us to diagnose a particular disease. So differential white blood cell count, sometimes just called DIF, is a count of the proportion of all the white blood cells. So remember we said that neutrophils make up, on average, in a healthy person, between 50 and 60% of the white blood cells. If we do a differential count and notice that, oh my gosh, there are 70 or 80% neutrophils, then we might be suspicious of a bacterial infection, especially bacterial or perhaps a fungal infection. An increase in the percentage of eosinophils frequently tells us that there may be a parasitic infection or that an allergic reaction is going on. A reduction in a specific type of T cell called a helper T cell, that would tell us that HIV infection may be a cause of what's going on here. So that looking at the increase or decrease in specific white blood cell counts can be a diagnostic tool for disease. All right, here we are at the last slide. We're going to discuss then a little bit about blood platelets, the thrombocytes. These are membrane-enclosed fragments of a cell, a specialized blood cell called a megakaryocyte. Megakaryocyte, because they have such a large nucleus. Karyo means kernel, so if we think about the nucleus as being the kernel of a cell, then it's got a really, really large nucleus. So here's a megakaryocyte right there with its really large nucleus. They develop from the hematopoietic stem cell in response to, you'll gotta believe it, it's a hormone. The hormone's called thrombopoietin. So you can think of it as like erythropoietin was the hormone that stimulated the production of red blood cells. Well, thrombopoietin is the hormone that stimulates the production of the megakaryocytes. These platelets will fragment off of the megakaryocyte, and so they'll do a little bit, so these may be some platelets that are forming here. Here we have a blood sample where you can see a clump of platelets that have all kind of clumped together, and here's a white blood cell in the middle of that clump. Platelets are capable of amoeboid motion, so they can crawl around like little amoebae, and they are their function is in hemostasis, in stopping the loss of blood from a cut blood vessel. So these platelet clumps will form. So if there is a cut, a vessel gets broken, then the platelets respond by becoming really, really sticky. Their cell membranes will adhere to protein fibers. They will adhere to each other. They'll adhere to the wall of the blood vessel right there at the cut site, and so they will plug up, as they adhere to the cut, and then the platelets adhere to other platelets. Then that will help to plug up a blood vessel wall, and they will also secrete chemicals, serotonin being the primary one, that will result in the constriction of the blood vessel. And they secrete other chemicals to help stimulate coagulation of blood. We'll talk about that toward the end of this chapter. So platelets will circulate something on the order of 10 days, and then they lose their function, and they are removed primarily in the spleen by phagocytes that are hanging out there. Normal count for platelets is somewhere between 150,000 and 450,000 of them per microliter. All right, there you have it. Everything you ever wanted to know about white blood cells and platelets, but we're afraid to ask. You know how to reach me. Send me an email, give me a call, drop by during office hours, catch me around class time. Look forward to seeing you soon.